Very early modulation of brain responses to neutral faces by a single prior association with an emotional context. Evidence from MEG. Morel et al. 2012. Abstract. Recent electrophysiological studies have demonstrated modulations of the very first stages of visual processing, less than 100 milliseconds, due to prior experience. This indicates an influence of a memory trace on the earliest stages of stimulus processing. Here we investigated if emotional, audio, verbal information associated with faces on first encounter can affect the very early responses to those faces on subsequent exposure. We recorded magnetoencephalographic MEG responses to neutral faces that had been previously associated with positive, happy, negative, angry, or neutral auditory, verbal, emotional contexts. Our results revealed a very early 30 to 60 milliseconds difference in the brain responses to the neutral faces according to the type of previously associated emotional context, with a clear dissociation between the faces previously associated with positive versus negative or neutral contexts. Source localization showed that two main regions were involved in this very early association effect: the bilateral ventral occipitotemporal regions and the right anterior medial temporal region. These results provide evidence that the memory trace of a face integrates positive emotional cues present in the context of prior encounter, and that this emotional memory can influence the very first stages of face processing. These experimental findings support the idea that face perception can be shaped by experience from its earliest stages and, in particular, through emotional association effects. Introduction. The way we perceive others changes through our different encounters with them. This is allowed by the flexibility of our visual system, which adaptively modifies the way it processes sensory stimulation according to prior experience. In particular, the emotional context in which a stimulus is encountered can markedly affect the memory trace in that stimulus. Recent neuroimaging studies have indeed shown that the emotional context associated with a neutral stimulus can influence the neural memory trace of that stimulus. This influence has been observed throughout the visual processing stream, including striate and extrastriate regions. In line with the view that memory and perceptual processing are underpinned by the same mechanisms and involve shared neuronal systems, it is, however, unknown whether this reflects modulations of the earliest stages of visual processing. May an emotional influence be observed from the very first stages of visual processing? Such a question can only be addressed using functional brain imaging methods such as electroencephalography, EEG, and magnetoencephalography, MEG, which allow one to follow brain activity on a millisecond time scale in healthy subjects. There is a growing body of evidence that the earliest visual evoked components in the time range of the so-called C1 in EEG or M70 in MEG, peaking around 60 to 90 milliseconds, and even earlier, do not reflect an invariant sensory processing, but may be influenced by various factors, including attention, categorization, and emotion. Very early modulations have also been observed in response to experience related factors. These modulations were initially considered as low level effects related either to iconic memory or to perceptual priming due to low level pictorial properties of the stimulus. But recently, early experience related effects that may not be explained by low level features have been reported, suggesting that the very first stages of visual processing may be influenced by prior experience. To date, only two electrophysiological studies have investigated the influence of the emotional context of prior experience on very early stages of stimulus processing. Using a classical conditioning paradigm, Stolarova et al. 2006 have reported a modulation of the C1 component in response to a simple grating stimulus that acquired an emotional value through its repeated association with an affective negative picture. This shows that the memory trace of a stimulus integrates emotional cues present in the context of successive encounters, and that these emotional memory components can modify the very first stages of sensory processing of that initially neutral stimulus. This is true at least for the very simple grating stimuli, which are best tuned to activate V1 orientation selective neurons. May the earliest processing stages of more complex biologically relevant stimuli, such as social stimuli, also be influenced by emotional association mechanisms? In support of this question, we showed in a previous study that face repetition priming effects differed according to the emotional expression of the faces, happy, fearful, or neutral, in a very early time range, from 40 to 50 milliseconds, and this after only one previous presentation. This suggests that emotional cues may modify the very early neural responses to faces. However, the use of facial expressions did not allow us to interpret our results as an effect of emotion per se, as the emotional cues were intrinsically part of the visual stimuli. For instance, the smile of the happy faces or wide open eyes of fearful faces. Our very early effect could thus be explained by low-level differences predictive of the emotional categories. The aim of the present study was to examine whether affective person information previously associated with neutral faces may impact the earliest processing stages, less than 90 milliseconds, of these faces. We used MEG, which combines both an excellent temporal and a good spatial resolving power, offering the unique possibility to study the spatial temporal dynamics of brain networks. Specifically, we examined the brain responses to neutral faces that had been previously associated with an extrinsic, auditory, verbal, emotionally laden context. We consider positive, happy, negative, angry, and neutral contexts and examine whether the brain responses to neutral faces may be influenced by a single prior association with these contexts in an early time range. This would imply that the very first stages of face processing can be shaped by the emotional context of a single prior encounter. In addition, modulations by either emotion or by a combination of emotion and prior experience have been shown in the time range of the M100 and M170, which index successive perceptual stages of face processing, although with somewhat inconsistent results. Thus, we also examined if an effect of the emotional context previously associated with otherwise neutral faces could be observed on the M100 and M170 components in response to the faces. Methods. Participants. 19 subjects, 8 female, participated in this study. All participants were healthy, right handed, had normal or corrected to normal vision, no hearing problems, and were native French speakers. They gave written informed consent for the procedure and were paid for their participation. The study was approved by the local ethics committee. One subject was subsequently excluded from analysis due to poor quality of the recordings, and the behavioral data from another subject could not be recorded for technical reasons. Thus, the behavioral results were analyzed on 17 subjects, 6 female, and the electrophysiological results on 18 subjects, 7 female. Stimuli. Face stimuli. 300 grayscale photographs of different individuals posing a neutral expression were gathered from 5 different sources: the Karolinska Directed Emotional Faces Set, KDEF, the AR Face Database, the NIM-STIM standardized face dataset, the neutral faces of Hernandez et al. 2009, and photographs from our laboratory. As the stimuli came from different datasets, they were first cropped similarly around the face outline and centered so that the eyes and nose fell at the same position across all pictures. All stimuli were then resized to 234 pixels of height with a width of 182 pixels and normalized in gray levels. Finally, the background color was set to black. 210 faces were used as the stimuli of interest and divided into three lists of 70 faces, with 35 male and 35 female faces in each list, to be paired with each type of emotional context: happy, H, angry, A, and neutral, N, as detailed below. Each face list was composed of stimuli from all five datasets in a similar number. We ensured that there was no significant difference in mean gray level across the three lists. Moreover, each list was associated with one of the three types of sentences: H, A, N. And this face sentence association was counterbalanced across subjects. For a given subject, the first face list was associated with happy emotional sentences, the second list to angry emotional sentences, and the third list to neutral sentences. Whereas for another subject, list 1 was associated with A sentences, list 2 with N sentences, and list 3 with age sentences, and so forth. The remaining 90 faces were used as target stimuli for the purpose of the test and were not included in the electrophysiological analysis. They did not differ from the stimuli of interest in mean gray level. Sentence stimuli. A corpus of verbal auditory sentences was created in order to be associated with the neutral faces. The initial corpus comprised 282 sentences with three types of semantic and prosodic content: 94 sentences with happy, 94 with angry, 94 with neutral content. The semantic and prosodic contents of the sentences were always congruent. The sentences were composed of 10 words on average. To ensure that the participants easily assess the meaning of every sentence, the most important word of each sentence was selected as frequent, more than 2,000, and highly imaginable, more than 2, based on the BrulX database. Although most of the sentences had a simple syntactic structure, 32% of the sentences per emotional category had an additional relative clause, including a second conjugated verb. The sentences were constructed with three different simple tense: present for 33%, present perfect for 39%, and simple past for 27% of the sentences for each emotional category. All this auditory material was pronounced by a female voice, but half of the sentences of each emotional category started with the personal pronoun he, il, in French, and were always paired with male faces. The other half started with the personal pronoun she, elle, in French, and was paired with female faces. The sentences were recorded by a single non-professional actress with the appropriate emotional prosody, for instance, congruent to the semantic content, in a soundproof room using the software Audacity. The sentences were digitized and normalized in amplitude. To select the final corpus of sentences, a pilot experiment was conducted with 10 participants having French as their mother tongue, 4 men. The participants were comfortably seated in a sound attenuated experimental room. They had to carefully listen to every sentence via earphones and to decide if the subject of the sentence experienced happiness, anger, or no particular emotion, neutral state. A fourth response choice was given if the sentence seemed impossible to classify in one of the three categories proposed or was incomprehensible. Response time was limited to 1 second. Answers were collected using a response pad linked to a computer equipped with E Prime 1.2 software. We then selected 210 sentences that were correctly classified by all the participants, 70 for each emotional category, 35 sentences with the personal pronoun he, and 35 with she. The mean number of words in the final sentence corpus did not differ for the three types of semantic and prosodic content. The difference in sentence duration reached significance. Post hoc tests showed no significant differences between the duration of angry and that of either happy or neutral sentences. However, there was a small difference, less than 100 milliseconds, between happy and neutral sentences. 24 remaining sentences from the initial corpus were used in association with the target faces. Procedure. Participants were comfortably seated inside a dimly lit, electromagnetically shielded MEG room in front of a translucent screen. Face stimuli were back projected onto the screen through a system including a video projector placed outside the room and two mirrors inside the MEG room. The auditory stimuli were delivered through plastic earphones placed in the subject's ears. Stimulus presentation was controlled by a computer equipped with an in-lab software and connected to the MEG data acquisition computer through a parallel port. 210 face stimuli were distributed in 7 blocks, each containing 30 different neutral faces, presented in a random order. The face lists 1, 2, and 3 were randomly distributed into the block so that each block contained 9 to 11 faces associated with each emotional condition, age, and NN. Each block was presented twice during the experiment with a different random order of stimuli within the blocks, and the block presentation order was varied between subjects according to a Latin square design. The blocks were repeated immediately or after one intervening block containing different stimulus so that the time interval between the first and the second presentation of the given face was comprised between 1 and 7 minutes, corresponding to 15 to 87 intervening items between the two presentations. On the first presentation of the blocks, the faces were associated with a sentence, whereas on the second presentation, the faces were presented in isolation. Thus, in the first presentation blocks, trials started with a fixation cross presented for a random duration between 600 to 800 milliseconds, followed by a neutral phase stimulus presented for 1500 milliseconds. The auditory sentence presentation started 500 milliseconds after the phase onset. Hence, the phase was presented in isolation for 500 milliseconds and in association with the sentence for the remaining 1000 milliseconds. The sentence presentation continued after the onset of the phase, followed by an intertrial interval varying randomly between 700 and 3577 milliseconds. In the presentation blocks, the trial sequence was the same as on first presentation, except that this time the neutral faces were presented alone and that the intertrial interval from the offset of the face to the onset of the next fixation cross varied randomly between 1500 and 2500 milliseconds. In addition, 2 to 5 target trials were included in every block for the purpose of the task, 6 to 14% of the trials. These trials were identical to the other trials of the block, except that 500 milliseconds after the face onset, for instance, at the onset of the sentence during the first presentation blocks, the face was replaced by the face of a different person. Subjects had to press a button with their right hand as quickly as possible whenever they detected such face change. This task was implicit with regard to emotional processing and ensured that the subjects paid attention to each individual face. The target trials were not included in the electrophysiological analysis. During the whole experiment, subjects were instructed to pay attention to both the faces and the sentences and were not informed about the face repetitions. They were also asked to maintain their gaze fixated on the center of the screen and to avoid blinking during the trials. MEG acquisition. The experiment was conducted on a 151 DC squid whole head MEG system at the MEG Center of the Centre de Neuroimagerie de Recherche CNR. CRICM up UPMC and CERM SNRS, Paris, France. Magnetoencephalographic signals were recorded continuously with a digitization rate of 1250 Hz, bandpass DC to 200 Hz. 17 external reference gradiometers and magnetometers were included to apply a synthetic third gradient to all MEG signals for ambient field correction. 3 small coils were attached to reference landmarks on the participant, left and right preauricular points, plus nasion, in order to check head position at the beginning of each block. Horizontal and vertical eye movements were monitored by 2 bipolar electrodes placed at the outer canthus of each eye and below and above the dominant eye. Electrocardiogram was also monitored by a bipolar electrode placed on the right collarbone and the left hip in order to monitor the subject's physiological state as well as to check for any artifact in the ERF responses due to cardiac activity and correct the MEG signal if necessary. In the present study, we did not apply any cardiac correction, as the visual examination of our data showed no contamination by the cardiac activity. In addition, the recording included the signal of a photodiode that detected the actual appearance of the face stimuli on the screen within the MEG room. This allowed correcting for the delay introduced by the video projector. 12 milliseconds, around 12 milliseconds, and averaging event-related magnetic fields, ERFs, precisely time-locked on the actual onset of the face stimulus. Trials with eye movements, eye blinks, or muscle artifact were rejected upon visual inspection of the MEG and electrooculogram signals. Event-related magnetic fields, ERFs, were then averaged for each condition between 200 milliseconds before and 500 milliseconds after the face onset. Data were baseline corrected according to the 200 milliseconds preceding face onset and digitally low pass filtered at 30 Hz. The mean number of trials included in the ERF averages did not differ across emotional conditions in the first and second presentations. Measures and statistical analysis. First, in order to examine the very early effect of prior emotional context on the magnetic responses to the faces, we performed mean amplitude measurements in 3 successive 30 milliseconds time windows from 0 to 90 milliseconds post-stimulus onset. Each time window, the mean amplitude of the activity was measured for each emotional context condition over a large group of 20 occipitotemporal sensors in each hemisphere. These were chosen upon visual examination of the topographical distribution of magnetic activities in order to cover the very first observed visual evoked component, namely the M70. The sensors of measurement included ml/RO21, ml/RO22, ml/RO32, ml/RO33, ml/RO42, ml/RO43, ml/RT12, ml/RT13, ml/RT22, ml/RT23, ml/RT24, ml/RT25, ml/RT26, ml/RT32, ml/RT33, ml/RT34, ml/RT35, ml/RT42, ml/RT43, and ml/RT44, where M is the first letter common to every magnetometer sensor. L/R means left/right. We measured the same sensors over both hemispheres. O and T indicate sensor over the occipital and temporal regions, respectively, and the final number identifies a given sensor. Second, we measured the M100 and M170 components in response to the faces under each condition of prior emotional context. The peak amplitude and latency of these components were measured in each participant on 3 occipitotemporal sensors that were fitted on the maximum of the component in each hemisphere between 90 and 150 milliseconds for the M100 and between 145 and 200 milliseconds for the M170. Except for one subject where the M170 peak was particularly late and measured around 250 milliseconds. This subject was not excluded from the analysis as he did not modify our results on the M170. As we were interested in the modulation of face processing by prior emotional context, we focused our statistical analysis on the second presentation of the faces. For each of the measured responses, the parameters measured over the selected sensors, mean amplitude, peak amplitude, or peak latency, were analyzed using univariate analysis of variance for repeated measures. With emotional context, happy, angry, or neutral, hemisphere, left or right, and sensors as a within subject factors. There was no modulation of emotional context or hemisphere effects by sensors. Thus, ERF data were collapsed across sensors, and we report the F and P of the ANOVAs with emotional context and hemisphere as within subject factors. When some significant effect of emotional context was found, we performed the same statistical analysis on first face presentation as a control, as well as an ANOVA including presentation, first or second, in addition to emotional context and hemisphere factors. This allowed us to ascertain that any effect observed on second presentation was indeed due to the association between the face and the emotional context realized during the first presentation. Greenhouse-Geiser correction was applied for the comparisons of more than 1 degree of freedom, and the Greenhouse-Geiser epsilon value for the adjustment of the degrees of freedom is then reported. Bose-Hock-Scheffes tests were performed to compare emotional context effects. All statistical analyses were performed using Statistica 8 software. Prior to the statistical analysis, MEG data were multiplied by -1 in the right hemisphere in order to compare the activities between hemispheres. This was done because the M70 topography is bilateral with a flowing in field in the right hemisphere corresponding to a flowing out field in the left hemisphere. A supplementary point-by-point ANOVA with emotional context as within subject factor was performed between -50 milliseconds and +200 milliseconds on each of the 151 MEG sensors, and for each time point for the first and the second phase presentation, respectively, in order to ensure that there was no other effect of emotional context in the early time range. Source localization. Source localization was performed with Brain Visa software in order to estimate the regions underlying the very early association effect. Cortical current source density mapping was obtained using a distributed source model consisting in more or less 13,000 current dipoles in each subject and condition. Dipole locations and orientations were constrained to the cortical mantle of a generic brain model built from the standard brain of the Montreal Neurological Institute using the Brain Suite software package. This head model was then warped to the standard geometry of the MEG sensor cap. MEG forward modeling was computed for each subject and emotional condition with the overlapping spheres analytical model. Cortical current maps were then computed from the MEG time series using a linear inverse estimator. The weighted minimum norm current estimate. These maps were averaged between 30 and 60 milliseconds for each subject and each emotional context condition under the first and second presentation of the faces respectively to obtain the mean current amplitude at each vertex of the segmented cortical surface in the time window of interest. An ANOVA with the emotion as within subject factor was performed on these mean activations at each vertex. For the first and the second phase presentation, respectively, only regions where at least 5 contiguous vertices reached a significant threshold of p equals 0.005 were considered as significant. Results. Behavioral results. Subjects detected the occasional changes of face identity occurring in target trials at ceiling, with a mean response time of 513 without any difference between first and second phase presentation blocks for both accuracy and response time. Event-related magnetic fields, ERFs. Several components classically elicited in visual paradigms could be identified in the ERFs, namely the M70 peaking at 75 milliseconds, the M100 peaking at 125 milliseconds, and the M170 peaking at 165 milliseconds. The very early effect of prior emotional context on face processing. According to our working hypothesis, we first focused our analysis in the first 90 milliseconds post stimulus onset in order to test if an effect of the previously associated emotional context could be observed on the brain responses to the faces in this early time range. Thus, we measured the mean amplitudes of ERFs in response to the second presentation of the faces in 3 consecutive 30 milliseconds time windows from 0 to 9 milliseconds. These measures were performed on a large set of 20 sensors in each hemisphere, covering the bilateral peak of the M70 under each emotional context condition. The ANOVAs performed in each time window with emotional context and hemisphere as factors revealed a significant effect of emotional context, together with an interaction between emotional context and hemisphere in the 30 to 60 millisecond time window. This reflected a marked effect of emotional context in the right hemisphere, while this effect failed to reach significance in the left hemisphere. As can be seen on figure 2c, this effect of emotional context was in the form of differentiated response to faces previously associated with a happy relative to an angry or neutral context. Post hoc tests confirmed that the effect of emotional context in the right hemisphere was due to a difference between faces previously associated with happy sentences and those previously associated with either angry or neutral sentences. There was no significant difference between angry and neutral faces. A similar pattern was observed in the left hemisphere, although the difference in mean ERF amplitudes for the faces associated with different emotional contexts did not reach significance. No significant effect of emotional context or interaction between emotional context and hemisphere could be observed in the other time windows, 0 to 30 milliseconds and 60 to 90 milliseconds. Could the effect of emotional context observed be due to some low level, non controlled difference between the faces associated with the different contexts? It is important to remind the reader that the association between the faces and the different types of emotional context was counterbalanced across subjects. Hence, this seems very unlikely. However, in order to check that no difference across our experimental conditions could be observed prior to the association between the faces and the emotional contexts, we analyzed the magnetic responses for the first presentation of the faces, where the faces were presented in isolation for 500 milliseconds before the onset of the associated sentences. We performed mean amplitude measurement on the same set of sensors and in the same time windows as during the second phase presentation. The ANOVAs run in each time window with emotional context and hemisphere as factors revealed neither any significant effect of emotional context nor any interaction between emotional context and hemisphere. Furthermore, the interaction between presentation, emotional context, and hemisphere showed a trend to significance between 30 and 60 milliseconds. This interaction reached significance when we refine our measurement to the 14 sensors in each hemisphere where the magnetic activity was maximum in this time window. Altogether, these analyses confirmed that the 30 to 60 milliseconds effect observed in the second phase presentation was indeed due to the association between the neutral faces and the emotional verbal contexts, and not to some low level visual differences between the faces. M100 and M170. The analysis performed on the amplitude and latency of the maximal peak of the M100 and M170 components revealed neither any significant effect of emotional context nor any interaction between emotional context and hemisphere. Finally, in order to check that there was no other effect of previously associated emotional context in the early time range, we ran a point by point ANOVA with emotional context as a within subject factor on every MAG sensor and every time point between minus 50 milliseconds and positive 200 milliseconds. This additional analysis confirmed the marked effect of emotional context on the brain responses to the second phase presentation between 30 and 60 milliseconds, as well as the absence of effect in the other time windows. The topography of the 30 to 60 millisecond effect overlapped with the sensors of measurement in our main analysis. This supported the view that the mean amplitude measurement performed, as detailed above, allowed capturing the emotional context effect appropriately. In addition, no such effect was observed during first phase presentation. Thus, our very early effect appeared as a robust effect of prior association between the neutral faces and the emotional verbal contexts that was well defined in both space and time. Source localization. We examine the brain regions involved in the very early 30 to 60 milliseconds differentiated response to the neutral faces as a function of the previously associated emotional context. To this aim, we estimated the cerebral sources of the magnetic responses to the faces in the second presentation blocks and computed the mean source amplitude in the 30 to 60 millisecond time window under each emotional context condition. The ANOVA performed on each vertex of the obtained mean cortical current maps revealed a significant effect of emotional context in two regions, the bilateral ventral, occipitotemporal, extrastriate regions, encompassing fusiform regions and the right anterior medial temporal regions surrounding the amygdala. The same source analysis on the first phase presentation did not reveal any region showing a significant effect of emotional context. Thus, the right anterior medial and the bilateral extra striate regions seem to be involved in the modulation of the very early neural responses to faces by the previously associated emotional context. Discussion. Our results show that the earliest stage of face processing can be modified by a single prior association with an emotional context. The magnetic responses to neutral faces differ between 30 and 60 milliseconds post face onset according to the type of emotional context previously associated with those faces. More precisely, the faces previously seen in a positive, happy emotional context evoked a dissociated neural response as compared to those previously seen in either a negative, angry, or a neutral context. Source localization showed that two main brain regions were involved in this very early effect: the bilateral ventral occipitotemporal extrastriate regions and the right anterior medial temporal regions. Our results add to a growing amount of studies reporting that prior experience can modulate the brain responses to a neutral stimulus in a very early time range. In particular, the very early latency of our effect is compatible with other studies that use face stimuli. Our results further show that these very early experience related effects can be influenced by the type of emotional context of prior experience. These results demonstrate that the very first stages of visual processing are not invariant, but already take into account prior emotion related experience with the stimulus, with dissociated responses to faces previously associated with a positive emotional context relative to the faces associated with a negative or neutral context. The early timing of our effect is in line with two previous studies in which investigated the influence of prior experience in relation to emotion. In a classical conditioning paradigm, Stalarova et al. 2006 observed modulations in the time range of the C1 component, 65 to 90 milliseconds, in response to gratings that had been repeatedly paired with a negative emotional versus a neutral picture. The authors concluded that acquiring an emotional value through the association between a neutral and an emotional stimulus may lead to a reorganization of the early sensory processing in the visual cortex. Furthermore, Morel et al. 2009 reported differential repetition effects between 40 and 50 milliseconds for faces with happy, fearful, and neutral expressions, suggesting the existence of very early emotional influences also when considering complex social stimuli, such as faces. Our results extend these findings in several ways. First, only neutral faces were used in the present study. The faces associated with the different types of emotional context were counterbalanced across subjects, and we tested the effect of the association of these faces with an extrinsic emotional context. Thus, our positive emotional association effect cannot be due to intrinsic physical differences between the face stimuli. Rather, it reflects emotional association mechanisms per se, which yielded very early differentiated responses to the faces on second presentation. Second, we used audio verbal emotional contexts, thus, in line with a recent behavioral study. Our results suggest a multisensory integration of auditory, contextual, and visual information in the memory trace of faces that can affect the earliest stage of face processing, even in the absence of the previously associated contextual information. Finally, Our results extend those of Stolarova et al. 2006 by showing that such early emotional association mechanisms can occur after only one association between a neutral stimulus and a positive emotional context, at least in the case of a highly biologically relevant stimulus such as faces. Note that these results do not indicate that all the information from the face or from the previously associated context is extracted within the first 60 milliseconds of face processing. The structural encoding of faces is typically considered to require at least, more or less, 170 milliseconds of processing. Our results, rather, reflect that the very first processing stages of faces, where basic information such as, for instance, line orientation or spatial frequency is extracted, may be influenced by some, possibly minimal, positive emotional contextual information. What can be the functional significance of such an early emotional association effect? This question is difficult to address here, as we did not record any behavioral responses to the face stimuli. Nonetheless, it is interesting to know that the emotional association effect was selective of the positive contexts, which described happy events. This may appear at odds with the widely accepted idea of a prioritized processing of negative, threat related stimuli. However, it is important to keep in mind that here emotion was conveyed by contextual information and was not inherent to the face stimuli. Studies on emotional associative memory have reported that neutral stimuli are better recognized if they were encoded in a positive relative to a negative or neutral context. This is explained by the fact that negative contexts are particularly salient. Hence, they may grab attention away from the associated neutral stimulus, resulting in abolishment of global emotional enhancement effect on memory. It may thus be suggested that the valence of our emotional contextual effect reflect a deeper encoding and slash or a richer memory trace for the faces encoded in a positive versus a negative or a neutral context. Alternatively, positive emotions in the context of social interaction may bear a particular relevance in humans, thus explaining why our effect was selective for faces associated with happy contexts. In line with this idea, one must keep in mind that our sentence stimuli described everyday life situations in which the positive contexts could have an important adaptive value. It is also possible that neutral faces tend to be perceived as slightly negative rather than purely neutral. In this case, the association between a neutral face and an emotionally positive context would constitute an incongruent or more ambiguous condition as compared to the association between a neutral face and a negative or a neutral context. Our results would then reflect an effect of emotional ambiguity or of emotional incongruence. Further experiments will be required to disentangle these alternative interpretations. In any case, it remains that a single prior association between neutral faces and extrinsic auditory emotional context during a simple task of face identity change detection resulted in a very early dissociation of the brain responses to the faces previously associated with the positive relative to the negative in neutral contexts. Source localization revealed two main cortical regions obtaining our very early positive emotion association effect: the ventral occipitotemporal extrastriate regions, including the fusiform gyrus, and the right anterior medial temporal regions, encompassing the amygdala. We underline that we perform source localization using a template of brain and not by taking into account each individual's brain anatomy. This limits the spatial resolving power of our source localization method, yet it has proven fruitful in past studies for shedding light on the brain sources of magnetic as well as electrical responses. Moreover, the regions involved are consistent with the social emotional nature of our stimuli. Indeed, several fMRI studies have observed effects related to the emotional memory mechanisms in early visual areas. Furthermore, the amygdala is at the core of a network that processes emotional stimuli of both negative and positive valence, and it also plays a central role in emotional memory. The fact that only right anterior medial temporal regions were implicated might indicate that the very early association effect obtained reflected implicit memory mechanisms. Indeed, previous research on either emotional memory and learning or on the perception of intrinsically emotional stimuli support the idea that the right amygdala may be preferentially involved in implicit and automatic processing of highly relevant stimuli, while the left amygdala would rather be involved in the explicit evaluation of emotional, mainly negative, events. Thus, the right lateralization of the anterior temporal activation observed in our study is consistent with the implicit nature of our task with regard to both emotional and memory processing. Most importantly, our MEG study revealed that the ventral occipitotemporal visual regions and anterior medial temporal region were involved as early as between 30 and 60 milliseconds. The latency of this effect may seem extremely early, yet electrophysiological studies have observed activity in the striate cortex beginning at 30 milliseconds. And by the time of the M70 peak, around 75 milliseconds, several areas in the visual pathway can already be activated. Similarly, the amygdala has been found to respond at very early latencies, as soon as 20 to 30 milliseconds. Thus, in line with these data, our results suggest that the extrastriate visual regions and the anterior medial temporal regions may be differentially activated by phases previously associated with different types of emotional contexts, as early as between 30 and 60 milliseconds. Previous research has revealed an influence of emotion or a combined influence of emotion and prior experience on face processing at slightly later time ranges, in particular between about 90 and 200 milliseconds, involving the M100 component, with associated early frontal, amygdalar, and occipitotemporal activities, and the M170 component, with associated fusiform activations. However, our effect was restricted to the 30 to 60 millisecond time window. These previous studies considered either the influence of emotion on face perception using mainly facial expressions, for instance, intrinsic to the face, or the influence of emotion on the explicit memory for neutral faces. Thus, the absence of emotional influence at later latencies in our study most probably results from the specificities of our paradigm that investigated the influence of extrinsic emotional context on the face on the memory trace of neutral faces using an incidental task. Altogether, our findings and those of previous studies support the view that the brain network involved in face processing depends on task requirements and processing goals, and that ventral, extrastriate, as well as anterior medial temporal regions may be involved in multiple stages of face processing. The very early time range of our effect may be taken as evidence for a very early reorganization of neural representations in the occipitotemporal visual cortices and the anterior medial temporal regions. These regions would then be reactivated very early on when the faces are viewed again alone. The existence of either a fast subcortical route towards the amygdala bypassing the primary visual cortex or of very fast feed forward cortical routes have been proposed, which may explain the very early co-activation of this structure with visual regions. Alternatively, it may be explained by very fast direct feedback influence from the amygdala onto the visual regions, as previously proposed. Further investigations will be needed to understand the neural routes obtaining the very early experience related and emotion related effects for which there is accumulating evidence. Conclusion. The present work shows that the very first stage of face processing between 30 and 60 milliseconds can be influenced by a single prior association with positive emotional contextual information. It further revealed that the bilateral occipitotemporal regions and the right anterior medial temporal region were involved in this very early effect of previously associated emotional context. Altogether, these results support the idea that face perception is a highly flexible process that can be influenced by the emotional memory trace from its earliest stages.